Hello students. Students, today we will discuss the structure of tRNA. tRNA, that is transfer RNA. We know that there are different types of RNA, rRNA, mRNA and tRNA. tRNA is about 10 to 15% of total RNA. It is synthesized in the nucleus. The synthesis of tRNA takes place inside the nucleus. It is also known as sRNA, that is soluble RNA. Why it is known as sRNA or soluble RNA? Because it is soluble in 1 m of NaCl. So that's why it is known as sRNA. It is the smallest RNA, that is 4S type. It is known as adapter RNA. Now why it is called as adapter RNA? Because it acts as a connecting link and bring the amino acid for the protein synthesis. So if the function of tRNA is asked, you can write down that it acts as a carrier of amino acid and it is required for the protein synthesis. So during protein synthesis, it acts as a carrier of amino acid. Now, here we can see the two-dimensional structure of tRNA, which was proposed by Holy. He compared it with the structure of a clover leaf. That's why the name is given clover leaf model. Clear? So this is the two-dimensional structure which was proposed by Holy. In your book, the three-dimensional structure is also given, so just go through it. Here we can see that it is appearing to be double-stranded, but actually it is single-stranded. Clear? So tRNA is single-stranded. Here hydrogen bonding takes place between the complementary nitrogenous bases and it looks like that it is double-stranded. Here we can see at the 3' end there is the nitrogenous basis CCA. Clear? This is the 3' end. And this end is known as acceptor end. Why it is known as acceptor end? Because the amino acid will bind at the 3' end. So that's why it is known as acceptor end. You have to remember the names of different loops and the ends. So 3' end is the acceptor end. It will accept the amino acid, or you can say the amino acid will bind at the 3' end. This is the 5' end where we can see guanine is present. Now what happens? There are 3 loops present here where the hydrogen bonding has not taken place. Here we can see when we go from 3' to this end, this loop is known as T-psi-C loop or T loop or attachment loop. So here we can see this loop is known as T-psi-C loop, attachment loop or T loop. So what is the function of this loop? The ribosome will bind to this site with the larger subunit. Clear? So it is the site where the ribosome will bind. This is the anticodon loop, or we can see the recognition loop. We will see its function later on. So here at the end we can see the anticodon loop. In my previous topic where I told you about the genetic code, I told you about the codon. So this is anticodon loop. This loop is known as D loop or DHU loop. Now we will see the function of each loop. So this is the structure and you have to remember it. It is very important from the exam point of view. First of all, we will see the acceptor end. Now, as I told you, at 3' end is the acceptor end. Amino acid binds to 3' end. Amino acid will bind at the 3' end because here we have the functional group OH, which makes it reactive by the COOH group present in the amino acid. So that's why it is known as acceptor end. Clear? Amino acid will bind by its COOH group. Next is the attachment loop. So this is our attachment loop, or it is also known as T loop or T-psi-C loop. See its function? It connects tRNA to the larger subunit of ribosome. We know that there are two subunits of ribosome, the smaller and the bigger one. Just see here, this is the smaller subunit and this is the bigger subunit. When the protein synthesis has to take place, what happens? These subunits unite together, and for that magnesium ion is required. So they remains free, but during the protein synthesis they will unite. So here we can say the smaller subunit and this is the larger. So here what will happen? Ribosome will bind with its larger subunit. It will bind to the ribosome with its larger subunit. Which loop? It is the T loop, or we can say the attachment loop. Connects tRNA to the larger subunit of ribosome. Next, here we can see the third one is anticodon loop. This is anticodon loop. Now what is its function? tRNA recognizes its place on messenger RNA. tRNA will go to the messenger RNA. It has to recognize its place and it will be done with the help of this loop, which is known as recognition loop, with the help of anticodon. There is a specific sequence of 3 nucleotides called anticodon at the end of this loop. Suppose we have a messenger RNA having the codon A, U, G. So this anticodon loop will have A. A will be having U, A and C. Clear? So there is a specific sequence of 3 nucleotides. So this is the 3 nucleotides called anticodon, which will be present at this anticodon loop, and this tRNA will recognize its place with the help of this anticodon loop. Next is DHU loop. So this is our DHU loop. It is also known as amino acyl synthetase recognition loop. During protein synthesis, during translation, an enzyme is required for activation of amino acid, and we have to remember the name of the enzyme. The enzyme is amino acyl synthetase. So this loop, which is known as DHU loop, it will recognize the amino acyl synthetase. So that's why it is known as amino acyl synthetase recognition loop. We know that there are 20 amino acids. There are 20 essential amino acids, so there will be 20 enzymes required for these 20 amino acids. DHU loop recognizes this specific enzyme. So this enzyme will be recognized by DHU loop. So these are the different loops. We have to remember the name of these loops and the function of these loops. We will just see once more. 3' end is known as acceptor end. It will accept the amino acid. Amino acid will bind at the 3' end. This is our T-RNA loop or attachment loop, which will get attached to the larger subunit of the ribosome. This is anticodon loop, which will help to find its place on the messenger RNA, and this is DHU loop, which will help to recognize the enzyme amino acyl synthetase. So this was all about tRNA. Thank you.